Konnichiwa guys, welcome to our channel Loops of Biology. I'm Yogiraj Jakkal, taking you all to the next part of the brainstorm. Now, as we have covered first of the brainstorm of this particular chapter Biomolecules, let's move on to the second one. Let's grow up your mind guys. So question, if enzyme A has lower Km, let's write it down, that Km value for enzyme A for enzyme A is low, and that of Km value for enzyme B is high. So now this is what the question is going to be saying. Then which enzyme is good and why? That's the main point. If I'm talking about good or bad enzyme, that is not decided by Km. Although the Km is going to tell me the affinity of enzyme towards substrate, it doesn't allow me to decide whether enzyme is good or bad. The complete good or bad of the enzyme is decided by the function of it. If I'm talking about, suppose, fermentation industry, if there is fermentation industry and I want to get my product as soon as possible. Alright, so in this particular case, the glycolysis, that is nothing but breakdown of glucose. That is nothing but breakdown of glucose has to take place at a faster rate. And it will require Km low enzyme. So in this particular case, enzyme A will be your good enzyme for this particular reaction. While if I'm talking about photosynthesis, where actually I'm going to synthesize glucose molecule, there will be synthesis of glucose molecule. And as the name suggests, photo means light. So the complete process is taking place within the light present in the surrounding. Alright. So this process is not going to occur whenever the light is completely shut down. So this is the process occurs only during the light. Got it guys? So in this particular case, the enzyme has to work throughout the particular daytime. In this case, the enzyme has to work throughout the daytime. And during this particular process, if the enzyme is working throughout the daytime, then its Km should be very, very high. That means affinity towards substrate should be very, very low. So in this particular case, Km high, that is nothing but enzyme B, is going to be my good enzyme. So the good or bad of the enzyme is completely dependent on the functionality of the enzyme. So from this, good or bad enzyme decides on functionality. That is nothing but when I am going to decide the function of the particular enzyme, where I am going to use that particular enzyme, that is going to be deciding factor for enzyme to be good or bad. Got my point guys? So this is your first question. Next is, let's try this particular question guys. If the substrate enzyme model of the enzyme was designed in such a way that it would be specific for transition state, then what would be the problem with that particular enzyme? So as we have seen two different models, those are nothing but, first one, lock and key model, lock and key model, and another one is nothing but induced fit model. Induced fit model. Okay. So in this particular case, in case of lock and key, it was very specific for the substrate. It was the enzyme is specific. Enzyme is specific for substrate. And that if I'm looking at induced fit model, the enzyme changes. The enzyme changes three-dimensional structure of active site based on substrate. Got my point? So in this particular case, we are designing one enzyme that is going to resemble the transition state of that particular substrate. Now, what do you mean by transition state? Whenever the substrate is basically moving to the product, it is occurring through an intermediate stage called as transition state. This is very highly energetic stage. So this is highly unstable and energetic. So it will create two different problems. If I'm talking about transition state, the enzyme will be specific for it. The enzyme needs to be specific for it. So there will be again a lock and key model problem. Alright, so there should be specific enzyme to be formed based on the transition state of that particular substrate. However, the transition state is highly unstable. Whenever the instability has been seen, that means the driving force towards stability is more, and due to which the transition state has to move from that particular unstable state to the stable state by formation of product. Got my point? So then that particular case, the transition state is stayed for a very short period of time. It remains for very short period of time. And it can go undetected. As well. So this will be two different problems which can cause whenever we are designing an enzyme based on the specific transition state of that particular reaction. Got it, guys? Got my logic behind it? And are you developing your logics behind it? Please comment in the comment box section. Next, if you are talking about Raj, alright, the Raj has been growing a bacteria for a long period of time. Okay. The bacteria is still in the growth. So there is a bacteria and they are forming bacterial colonies on a medium. Alright. That medium was containing Mg2+. That medium was first containing Mg2+. Now that is completely switched by Zn2+. Now this Mg2+ is now completely switched to Zn2+. Then what will be the problem? That is my question. Alright. In that case, they have given you the problem. They have asked what is a possible reason behind it. Alright. The growth of bacteria has been seen to reduced and completely stopped. So at the end, bacteria has stopped growing. Got it, guys? So now why is this happening? We have to figure this out. Whenever the bacteria is grown in the medium containing Mg2 plus, that means this ions enters inside bacteria, enters in the bacterial cell, and they are specific. They should be, or they are specific. So they should be specific. They should be specific for enzyme regulating growth. Specific for enzyme regulating growth. That means there has to be cell division to take place. There has to be cell division to take place, and due to which the growth is positive. Due to which the growth was positive. Alright. So in this case, Mg2 plus was a perfect cofactor. Was a perfect cofactor. And that was allowing active site. And that was allowing active site to attach to substrate. Attach to substrate. Alright guys. So whenever we are changing that to Zn2 plus, then the thing is changing that the type of cofactor is changed. That means the active site will not be open. The active site is close, due to which no substrate binding, and that will stop this particular growth of bacteria. So cofactor is going to play an important role. The cofactor is going to play an important role whenever the enzyme functioning is going to be decided. As the bacterial colony has stopped growing, that means their enzymes are not working at their proper pace and at a proper position. Whenever we are not allowing enzyme to work yet at their proper pace, that means bacteria has to stop growing at the end, because they are not achieving as much as energy which is required for the cell division. So that's why enzyme functioning is very, very important, and the activity of enzyme is governed by one of the factor as cofactor. Which is nothing but your metallic ion, and that metallic ion has to be Mg2 plus or Zn2 plus. Got my point, guys? So Raj has replaced this Mg2 plus to Zn2 plus, due to which the cofactor has been completely changed, and due to that particular change in the cofactor, the active site remain close and there will be no attachment of the substrate, and then the bacteria won't have enough energy for the cell division to perform, and it will stop growing that particular pace. Alright. Let's move on to the next question. That is, Rashmika wants to know the role of gene A. Alright, if you are talking about gene, that means there has to be a topic related to nucleic acid. Alright, so this is a part of nucleic acid. So gene A in a bacterial DNA, then what can be the possible experiment approach should she take to find it out? Alright, if we are talking about the functioning of gene, that means we are deciding to stop one of the metabolic pathway. Alright, so nucleic acid, whenever we are talking about role of gene, it can be A, it can be B, whatever. Whenever the role of gene is considered, that means we are halting metabolic pathway. And that is one of the important things. Whenever we are going to halt the metabolic pathway, that means we have to stop the protein production. That means there should be stopping off protein production. protein production. As we are back tracing this particular protein production, this protein is coming out. Protein is coming out. Protein is coming out from mRNA, and this mRNA was obtained from your DNA. Alright. So this process is basically working in this particular direction. That means DNA to mRNA, mRNA to protein in this particular case. Got it, guys? So whenever I'm talking about protein formation, that means I have to stop mRNA gene to express. Alright. mRNA gene to express. So this mRNA should be stopped expressing. Stopped expressing. And this stopping is basically carried out by introducing, so there should be introduction of microRNA to completely remove that particular gene, or there can be small nuclear RNA, or that is called a small interfering RNA, that is SN or siRNA, that will silence that particular gene. That will silence that particular gene. That means there will be the protein production will stop, but for a while. And during the stopping of protein production, whatever the phenotypic changes are taking place within the bacteria, that will decide the role of particular gene. So after stopping the gene expression, the phenotypic changes are going to decide the role of gene, and that is very, very important. So whenever we have to discard any metabolic pathway or understand the role of gene inside the bacteria or human cell or any other organism, then we have to stop that particular protein production by stopping the expression of mRNA, by using miRNA or snRNA, and finally look for the phenotypic changes that will clear you out whether this particular gene is working for good of the cell or bad of the cell, or in which of the stages of metabolic pathways. So you can understand everything whenever you are going to halt that particular protein production. Got my point? And if you want to see the working of that particular gene, where that particular gene product is basically working. So if the site of product to work in the cell has to change, then you can add radioactive tag on that particular product. That means there can be either fluorescent protein can be formed. That fluorescent protein will give you an information regarding the site of that particular product working inside a cell. Got it, guys? So this was a particular experimental setup. Now, some organisms are seen to be adapted at higher temperature. What can be the mechanism behind it? Whenever I am talking about higher temperature adaptation, all right, so adaptation is one of the skill where there can be mutations within DNA. Mutations within DNA can occur. All right, the surroundings of that particular organism is going to decide what is going to be the sequence of DNA. And that sequence, if the sequence is changed, the sequence should be changed. As soon as the sequence is going to change, that are going to make thermostable protein. So there should be thermostable protein. Now why I am calling it as protein? Because the proteins are degraded at a faster rate, as we have seen that in the graph of enzyme. If not, just go and watch my video on effect of temperature on enzyme. Whenever I'm talking about thermostable protein, that means the protein can withstand withstand at higher temperature as well. Withstand at higher temperature as well. So whenever I am talking about the organisms which are staying at a higher temperature, they are making thermostable protein and they are protecting their cell membrane by allowing this protein to cover them up. All right. So there will be the proteins helps to cover and protect cell membrane. Cover and protect cell membrane. So thermostable protein is one of the key concept. Thermostable protein is one of the key concept you have to learn in this particular part. So this is the new concept, guys. Got it? So these are few of the questions of the brainstorm. I hope you love this video and you are developing the logic behind the biology. These two chapters were very, very important and the base of biology, without which you cannot build your actually biology building in the future. Got it, guys? So be focused and be well versed with these two chapters. And I hope you are going through every of my videos so that you will be able to develop the logic properly in the subject biology. Okay? So please do not forget to like, share and subscribe my channel. Hope to see you in the next video. Bye bye.